Welcome back to MLT 240. This is part 2 on cutaneous mucosa or fungi. Some of the species that we're going to discuss include Microsporum, Epidermophyton, and Trichophyton. Some general characteristics of our cutaneous mucosa. These are referred to as dermatophytes. These are fungus that are able to use the keratin and affect the hair, skin, or nails. They usually live freely in the environment. A few species have adapted to live mostly exclusively on living animals, and a few on human tissues. Typically, they form two sizes of reproductive cells, macroconidium and microconidium. These are asexual conidia. They have distinctive sizes, shapes, and surfaces that help with species ID. Infections in humans are rarely inflammatory. Species ID in anthropophilic species can be difficult because most produce very few conidia. Here is a list of dermatophytosis based on body site. Tinea is also referred to sometimes simply as ringworm. There's Tinea captus that affects the scalp. There's Tinea corpus that can be anywhere on the body. There's Tinea barbae that can be in the beard. It's also known as barber's itch. Tinea curis that affects the groin. It's also known as jock itch. There's Tinea pedis that affects the feet. It's also known as athlete's foot. And finally, we have Tinea unguium that affects the nails. Now we're going to go on and look at some images of each of these on the different body sites. This is an image of Tinea captus on the scalp, and you can see it right here. This is Tinea corpus, and this looks like it's on the stomach area right above the belly button. This is Tinea barbae or barber's itch. This is Tinea curis or jock itch. This is Tinea pedis or athlete's foot. And finally, this is Tinea inguinum that affects the nails. For lab ID for dermatophytosis, of course, we can use our KOH preps. The calicofluor white can enhance detection through fluorescence. Arthrocandidia in the hairs can also be used to determine IDs and help speciate. Endothorax and ectothorax can also be used to help indicate a dermatophytosis infection. This is indication of inflammation either inside the hair shaft or outside the hair shaft. If there's no fluorescence, it could indicate endothorax or a infection inside the hair shaft. If there's fluorescence, it can indicate ectothorax or a infection on the outside of the hair shaft. In lab, you could also do a dermatophyte test medium or a DTM. It's useful as a screening medium. After 14 days incubation at room temperature, the yellow indicator changes to red if an organism is a dermatophyte. You could also use the Sabros brain infusion with cyclohexamide and chloramphenicol as an isolate for dermatophytes. And you could also use slide cultures to speciate. Certain species are more likely found on certain body sites than others. Microsporum are more likely found on the skin, scalp, beard, and body. Epidermophyton species are more likely found on the skin, the feet, the groin, and the nails. Trichophyton species are more likely found on the nails, groin, feet, the beard, the body, and the scalp. A small handheld UV light known as the wood lamp can be used to detect certain bacteria, fungi, and infections. In infected areas, that area will fluoresce, and a lot of times the species that are found is Microsporum species. And there's different growth rates for different species. Some growth rates are intermediate, between 6 and 10 days, and some species are slow growers, between 11 and 21 days. Here are some dermatophyte microscopic characteristics by genus. For Microsporum, they produce numerous rough, thick, or thin-walled, elliptical or spindle-shaped macroconidia containing 3 to 7 cells. A few club-shaped microconidia singly along the hyphae. Macrosporidium and Dewey rarely produces macro or microconidia. For the Epidermophyton species, the Floccusum is the only pathogenic species that develops numerous smooth, thin-walled, club-shaped macroconidia containing 2 to 4 cells. No microconidia. For the Trichophyton species, they usually form thin-walled, pencil-shaped macroconidia with 3 to 8 cells. Some strains produce numerous macroconidia. Numerous round, oval, or club-shaped microconidia in grape-like clusters or singly can be along the hyphae. T. schulani and T. violaceum are exceptions. T. schulani are known to show a fabric chandelier formation under the scope. For Microsporum species, all species can cause ectothrax or infection on the outside of the shaft of the hair and skin invasion. For M. canis, it causes a fluffy white colony with bright yellow on the reverse pigment of the plate. The macroconidia is tapered, elongated, and spindle-shaped. You have abundant club-shaped microconidia attached to hyphae. This species is usually contracted from either dogs or cats. For M. andunii, this is a species that rarely produce conidia on culture, and the colonies are fluffy white with little or no reverse pigment on the media plate. Here's a look at what microsporum can look like under the microscope. These larger structures are the macroconidia. This is an example of microsporum on a media plate. This is the top side, the fluffy white colony, and this is the reverse side as in M. canis, and you see the yellow pigment on the underside or the reverse of the plate. E. floccusum is the only pathogen in this genus, and it causes athlete's foot. It's contracted from person to person and is distributed worldwide. It affects the skin and the nails, but not the hair. On microscopic exam, it produces only one size conidia, and it's described as a microconidia because of the size, and it's produced in clusters or singly. It's on the distal end of the conidium and is broad or spatulate or said to look like a beaver's tail. Occasionally, conidia can be single-celled, but usually are separated in 2 to 5 cells by perpendicular cross walls. This species is notorious for developing pleomorphic tufts of sterile hyphae in older cultures. Here's what Epidermophyton looks like under the scope. You can see the telltale macroconidia that looks like beaver tails. Here is a culture plate growing a colony of Epidermophyton. Trichophytons is the last species we're going to discuss on this portion of the lecture. In lab, it's sometimes necessary to differentiate between the two species of T. menagrophytes and T. rubrum. In the first species, T. menagrophytes, it's the most common cause of athlete's foot, and the red strains may be confused with T. rubrum. You can use urase to differentiate between T. rubrum. T. rubrum is going to be negative, where T. menagrophytes is going to be positive with urase. T. rubrum is a deep red reverse pigment on the plate for growth. T. rubrum causes dermatophytosis in the body, the feet, the groin, and the nails. In patients receiving immunosuppressive therapy, T. rubrum may cause subcutaneous nodules or abscesses. Here's an example of Trichophyton on a media plate. You can see the fluffy white colonies on the front side of the plate, and you can see the dark red pigmentation of the colonies on the reverse side of the plate, and this is after 2 weeks of growth. Another species of Trichophyton that you might run across in lab and that affects patients is Trichophyton transurans. It causes 90% of Tinea captus in the U.S. It's also called black dot Tinea captus. It causes endothrex or infection inside the hair shaft. The colonies form a rust-colored color or pigmentation on the reverse side of the media plate. Here's what trichophyton can look like under the microscope. Here are some media plates of different species of trichophyton. These top images are of the top or the front side of the media plate, and these bottom images are of the reverse side, showing the pigmentation on the bottom of the plate. This concludes part 2 of this lecture. I will see you over in part 3.